The thing about our strategy system is that it's just like super powerful and can really help you form hypotheses and test those hypotheses to direct your lab work. Most people here are molecular parasitologists, right? And Omar has already given a bit of a demo about that, but I wanted to go into a little bit more depth about the data and how that is used in our searches, and to talk about how we combine lists from your results in your strategies and what's that used for. So the best way I know to do that is to go to a gene page. So I'm going to, here I'm in PlasmaDB. I entered an ID in the site search, and there was an exact match in the database. So I can use that link to get to the gene page for apical membrane antigen 1. And this gene that I'm looking at right here, this gene record belongs or came from the P. falciparum 3D7 genome. So there are other apical membrane antigen genes in our database, but I'm specifically looking at the one from the PF3D7 genome. So let's talk a little bit about the genomes and where they come from. We actually are not the owners of these genomes. We download them from the NIH or other INSDC repositories. So we download the full genome and the full annotation. So we're not looking at specific sequences that individual researchers have uploaded to GenBank. We're actually downloading the full genome. And as we download the genome, we run bioinformatic analyses on it. So, well, let's talk about what we get from the genome sequence and annotation. We get the gene models. So, and we call that gene structure, right? So we know if a gene is annotated to have UTRs, how many exons it has, that type of information from the genome. We take all of our genomes and run them through OrthoMCL. So we compare, OrthoMCL is an ortholog, is an algorithm that will take all of the protein sequences, again, and blast them against all of the other protein sequences across all of the proteomes that you give it. So we do this on all of our organisms, which we have over 600 of them now. And then what you get back is then when you cluster that reciprocal best hits, and you come up with clusters of genes that are orthologs, right? And so we use that on our sites to define orthology and to do these transformations that you've seen Omar do already, right? So if you go down to the orthology and synteny section, you can see how many apical membrane antigens there are within PlasmaDB and how many and what other organisms have that, right? We also have a lot of transcriptomic data. So for not only do we bring the genome sequence and annotation in from the INSDC, we also bring in raw RNA sequence data, and we align that to appropriate genomes and come up with expression data. Now, what's a little bit different from what we have from the publication is that we have used the same workflow to analyze all of these different experiments. So when Zhang et al. Looked at their microarray data, they applied a certain analysis that helped them analyze the specific question that they wanted to analyze. But since we're sort of like pan bioinformatics, right, we want to be able to compare all of these experiments to each other. So we use the same workflow on all of the data. And what we produce then is a table of each gene and its expression value within the samples that defined this, this transcriptomics data. We also have proteomics data in a similar fashion. We receive peptides from researchers and are able to map them back to the genome. And so we have information about whether this gene is expressed at the protein level. Not only do we get peptides here, we also get quantitative information. So an abundance could be an absolute abundance or a relative abundance based on, a published experiment piece at all in here. So we have a ton of information, ton of data, and not only do we have this data for this gene, AMA1, but we have it for every gene in this genome. And we have it for every genome in this database, PlasmoDB, and we have it for every genome in all of our other databases too, about 600 organisms now. And so imagine if you were a computer scientist, you could write a script that pulls out specific portions of this data. You could say, show me all genes that have 2 exons. Show me all genes that are expressed at a certain, life cycle stage. Right, and that's what our searches are. And then we package that into an interface where hopefully it's easy to use and easy to access that. You don't have to write any, you don't have to write any code or anything like that. But you can still pull out genes that are differentially expressed based on that data. Okay, and then I'm going to go up here to my strategies. So we have individual searches, and each one of these is the results of an individual search, but you can also string them together. And we offer Boolean operations that combine the lists returned by each search into a new result. And that's called actually a step result. So, and we call this a strategy. In fact, we call just one search a strategy, which is just single strategy with a single step. Okay, so I'm going to run a couple searches now and go into a little bit more depth about how to combine these searches. All right, so say that I want genes that have transmembrane domains and signal peptides. They have to have 2 biological characteristics. One is a transmembrane domain, and that same gene needs to have a signal peptide. So I'm going to go to searches and look for transmembrane domains, count. Again, I'm going to look for genes that have between 1 and 99 transmembrane domains. And I find that there are 87,000, almost 88,000 genes of this type that have at least one transmembrane domain. And if I look at my results, up here is the growing strategy. I have an organism filter on the left where I can, say I only want to look at P. falciparum 3b7. And of those 88,000, there's 1,600 that are in P. falciparum. And then on the right here it's just a list of all of the hits, right? These are all of the genes, and I can, go to certain gene pages if I look at this, use this link here. But say I change my mind, and this is the power of in silico experimentation, that I really want to only find genes that have 3 transmembrane domains, right? So I can do that very quickly, very easily. I can click edit here, revise, and change my parameters. It has to have a minimum of 3 transmembrane domains and a maximum of 3 transmembrane domains. And I find that there are only 127 of these in P. falciparum. That little red filter there is to remind you that you have taken the results and said, show me only P7. Okay, but so our original quest really was for genes that have a transmembrane domain and a signal peptide. And of these 127 genes that we're looking at, we know that they have 3 transmembrane domains, but we don't know if or how many signal peptide domains they have. Well, I take that back. The gene would only have 1 signal peptide, I believe. Yeah, so we don't know. We know these have transmembrane domains, but we don't know if they have signal peptides. So I'm going to use my power of in silico searching to say add step. And then I have a couple choices, and I'm going to choose to combine my transmembrane domain genes with the results of another search. And I have to choose how to combine these 2 lists of genes that I'm going to get, and I have to choose the search, right? So let me digress just a little bit and talk about how to combine these 2 lists that we're going to get. All right, so our search 1 results that we just did is returns transmembrane domains, and our search 2 is going to return a list of signal peptide domain containing genes. If I intersect these, I'm asking the question, show me all of the genes that are in common between these 2 lists. So they have to have a transmembrane domain and they have to have a signal peptide. If I union these 2 lists, I'm just going to say, show me all of these genes. Give me a list of all of these genes. And so the biological properties of our result list will be a little bit different, right? We will have genes with transmembrane domains, genes with signal peptides, and then some of those will have both, right? So that's asking a little bit different question than if you intersected these genes. And then you can say 1 minus 2, so show me transmembrane domain genes, domain containing genes that do not have signal peptides, or vice versa. And then we also have this 1 relative to 2, which I will show you in a little bit. Let's not do that right now. Okay, so I'm going to choose to intersect, right? Because we want transmembrane genes and signal peptides. And I'm going to run my signal peptide search here. Select all orders. It doesn't have very many parameters, right? And I'm going to say run step. And I come up with 29 genes that we know have transmembrane domains and signal peptides. Okay, so I come back from a seminar, and that was all about variation and single nucleotide polymorphisms. And I think, well, you know, it'd really be nice to know how many of these 29 genes have single nucleotide polymorphisms, you know, upstream or something. So it turns out that we actually, that, you know, some of the data that we have addresses single nucleotide polymorphisms, and that is data where folks have gone to the field, collected isolates, gone back, sequenced all of those different isolates, and then we have calculated, we've compared that to the P. falciparum 3D7 genome and called single nucleotide polymorphism. So wherever there's a mismatch between the isolate sequence and the official genome sequence, then we call that a single nucleotide polymorphism, right? So I'm going to go ahead and say add step. And the thing is, when I do a search for single nucleotide polymorphisms, they're not going to be genes, are they? I'm going to pull up, when I do that search, I'm going to pull up records for SNPs. So that's a little bit different beast to deal with. Right, so let's say we have our genes from, actually we're at step 2 now, and we know these are TM and single peptide genes, but we're going to return a list of single nucleotide polymorphism records. And so if we wanted to intersect those, we would say, how many of these single nucleotide polymorphism records are in this list of genes, you know, and that's just like comparing apples and oranges. But the thing that we do know about these both is their location on the genome. So we created this collocation tool to help you interact with this data. And the question you're going to ask is, how many of these single nucleotide polymorphisms are within a certain distance, or, you know, contained within or upstream or downstream of these genes? It's, again, a super powerful tool. So we don't want to combine with other genes. We want to do a genomic collocation, looking for single nucleotide polymorphisms, and I'm going to do differences within a group. Need to make sure that I have the accessing isolates from the organism that I want. I'm just going to go ahead and say, yeah, show me SNPs from that are, you know, use all of these isolates when you show me SNPs. And I'm going to change this to 50. Now you've told it what search you want to run. Now it's asking you where are these single nucleotide polymorphisms? Define the relationship between where the genes are and where the SNPs are. So you have to complete this little sentence, return each gene from the current step whose upstream region overlaps the exact region of a SNP and is on either strand. I'm happy with that. I'm going to say run step. Might take a while to finish. And what I'm going to come up with then is a list of genes that show, okay, so it turns out that 26 of those genes have upstream single nucleotide polymorphisms. I hope that gives you an overview of the nuances of combining searches and how to combine records or different features of the genome when the math behind it doesn't really work, right? You can't subtract lists of SNP record IDs from lists of gene IDs. There is, we have a few minutes. There's also another tool that the strategy system has that is really, really wonderful and easy, great to use, because you want to take advantage of the breadth of data that is available in all of our sites. So say I have these, this massive strategy up here, right? And you've looked for, you've unioned this information about, probably about a Go term. So if you looked for, you've defined proteases here or something, and then you used an RNA-seq data set to define, well, let's look at it. Yeah, so we've used an RNA sequence data set to look for expression in gametocytes. And we found that there were 1,400 genes like that, and the intersection of these protease genes with that is 400. And then we go through and we look at variation, and we come up with these 234 genes that are, fantastic on our list. And we're like, well, there's really a lot more data available about gametocyte expression on PlasmoDB. So how do I add that into this strategy? And the answer is to create a nested strategy, right? I'm going to click edit and make a nested strategy. And what shows up is something along the bottom here that begins with our RNA-seq gametocytes, but has the option to add another step here. So I'm going to say add a step, and I'm going to look for other genes based on the RNA sequence data. And I'm going to say, I want to search for, I'm going to find another experiment that has gametocyte data. And make sure that that is in p-falciparum PD7. So I have these gametocyte transcriptomes that I'm going to look to see if look for genes that are highly expressed in those samples. Male and female doesn't bother me. I'm going to look for the top 20th percentile of those genes and say run the step. Okay, so what we did here was we intersected our two gametocyte searches, and one returned about 1,500, the other one returned a little over 1,700, and the intersection of those is 981 genes, which is caused the final result to be restricted. There used to be 234 genes here, and now there are 149 because we're being a little bit more strict with our expression data. We're saying, return me only genes that are expressed, that were found to be expressed in gametocytes in both of these experiments, right? So that's a little bit more strict. We can broaden this and say, change the operand here, click edit to union these two RNA-seq data sets results and click revise. Yeah. And now we have 2,203 genes that are in common, that are, the non-redundant list of both of these search results. And our final result went from 234, I think, to 265. So we've actually identified a few more genes that possibly, that were found to have gametocyte expression in at least one experiment. And you can go on and add all of the RNAs. Do that if you like. But I hope I've gone over a few of the nuances of combining genes and what the data means and how we access it. I'll go back to the schedule. Were there any questions?
I think we answered most of them online.
Okay,
Just quickly, there were some questions about methods, like what method is used for determining signal peptides or transmembrane domains. And we do have a link to our method section, which includes sort of lists of algorithms that we use. Just one note is that we also, in addition to algorithms that we run, we also run our genomes through the EBI genome pipeline. Suzanne is showing you what that looks like. And so some of the links here actually take you out to EBI's processes. So that's kind of a nice advantage that we work closely with them and they run it through their pipelines. And then, we have to look through those to figure out exactly what version of the software they used in the latest release.
Yeah, this is really just an overview of what we do, our methods.
And then one note, there was a question about gene models. And of course, whether, can I find genes that have only one exon or two exons and so forth. And the answer was yes, we have a search for that. And the only thing I was going to say is anytime you're trying to do something on the resource and you're finding yourself sort of repeatedly doing the same thing over and over again, send us an email and ask if there's a way to do something more automatically, because sometimes there is. We might have a specific search that really will instead of you having to walk through every single gene page and look at the gene models and figure out how many exons they have, or download a table and then parse that, we may have a search that allows you to just quickly get it in a search strategy. And in this case, we do. So definitely drop us an email to ask about it.
Omar, there are 2 additional questions in the chat now.
So the question about, so there's a question from Juan about, is it possible to filter out group of genes that have common cis regulatory elements? So I don't know, we don't have that, I guess, per se. So it depends on the database. I know in class.
Yeah, if your common, what you're looking for can be defined as a motif, we do have, ways to do that. But if it's an undefined cis element, then you're we don't really have a way to do
right.
that. We don't.
Yeah.
Yeah.
So that's a good point. So you can configure a specific motif if you have an idea about the motif. And in fact, the motif, you can use something called the regular expression and be a bit more loose about how you define it, which is quite useful. Let's say you want to define genes that have, or you want to find motifs that have a T, a G, a C, followed by anything, followed by a G or a C, and so forth. So you can create a regular expression like that and find it. And then Suzanne briefly mentioned the collocation query. So then you can collocate the motif that you found with genes. So you can say, well, now return all the genes that are within 500 nucleotides of the motif that I discovered. So you can do that this way. And then I know in PlasmoDB, we have, for example, specific AP2s where you can select the AP2 and find the genes where those AP2s bound the upstream regions. That's based on a particular study, but that's really specific to that. And then some databases, we have some ChIP-seq data that's available for searches, which may be useful also for defining some maybe commonly regulated genes. And then again, not exactly that question you asked, but we also, for some of our databases, depending on the data type, we have genes that come from expression networks. And so you can define genes based on the expression network they fell into. And so again, as a surrogate, maybe, I know that in FungiDB we have that, so you can find genes that are co-regulated based on hundreds of experiments, and then you can potentially go from there. Hopefully that helps.
Omar, there is another question that asks, what is our relationship with the versions of Ncbi genomes?
Yeah, so this is Jesse. Do you want to take the lead on answering that question, since you're involved in that working group and you've been communicating with them? Or I'm happy to do it either way.
I'm happy to, but I'm wondering if we should take it offline and get started
Sure.
in the breakout rooms, and then I'm happy. Perhaps Rachel, you can put Lois and I in a room together for a minute, and then I'll join my regular group.
Sure.
Sounds good. So we probably should start the breakout room, Suzanne. Is that, or were you going to do anything else?
Oh, I thought Rachel would do that.
Yeah, no, I mean, before she puts us in case you had some other thing you were going to cover, but yeah.
Oh, no, that's fine. I don't have anything.
Okay, perfect.
We have, well, actually, we have two exercises. The top one called search strategies has less explanation than the search strategies homework that has more explanation. So I suggest doing the top one today during class, and it should be quick to go through that. But if you are interested in learning a little bit more about the strategies and need a little bit more help than the homework, might be able to give that to you. Yeah.
And then the goal is we'll go for about 40 minutes or so, and
Okay.
then we'll have a sort of a wrap-up discussion in the group, answer any general questions, and then as the schedule indicates, you're free to go at the top of the hour. We're not going to hold you to stay, but some of us will hang around for another half an hour in case you have questions, and
Right.
you're welcome to stay on and ask those questions. Okay.
And I'm sorry.
Yeah.
I thought
Also, since many people are interested in the answer to the NCBI question, I'll answer it at the end of the workshop today, so we don't have to worry about special rooms and everyone can hear it if they want to.
Okay, perfect. So Rachel, we're good to go into breakout rooms.
All right.
Rachel, you can probably stop the recording.
Thank you for the reminder.
I just noticed
Just a
it.
second.